Hi dear students, welcome to another session on solid waste management. And in this session we are going to discuss about anaerobic digesters and methanogenesis. First let's see about anaerobic digesters. So what is anaerobic digestion? Anaerobic digestion is the natural process that breaks down organic matter in the absence of oxygen to release a gas known as biogas, leaving an organic residue called digestate. Biogas is a mixture of methane, carbon dioxide and water and can be used to produce electricity and heat, or it can be used as a natural gas substitute. Digestate is a nutrient-rich byproduct of anaerobic digestion and can be used as a fertilizer and soil improver. In many countries, waste is badly managed and in some cases not at all. Poorly or unmanaged waste is unsightly, damages the environment, impacts the human health and attracts flies and vermin. Improper landfill disposal is a favorable and unsustainable waste management method. Anaerobic digestion offers a more sustainable waste management option, recovering resources and diverting them from landfill disposal and uses less space than landfills. Uncontrolled anaerobic digestion in landfills and waste dumps releases greenhouse gas emissions including methane and carbon dioxide. Controlled anaerobic digestion systems can capture methane for use as a valuable renewable fuel. Capturing the biogas reduces greenhouse gas GGH in two ways. The first one way is through preventing uncontrolled methane emission, and the next is by generating energy that displaces the use of fossil fuels. Anaerobic digestion is a regulated version of natural events of landfill in that it results in the controlled release of methane-rich biogas, which offers the potential for a very real form of energy from waste. And anaerobic digestion is carried out in large fermented tanks. In these tanks, solid waste is taken in the absence of oxygen and the anaerobic bacteria convert the large organic molecules mainly into methane and carbon dioxide. Unlike composting, anaerobic digestion occurs at one of three distinct temperature ranges, namely cryophilic, which is less than 20 degrees Celsius, mesophilic between 20 and 45 degrees Celsius, and thermophilic, which is greater than 45 degrees Celsius. Microorganisms involved in anaerobic digestion can be classified into four main groups. The first one is hydrolytic fermentative bacteria that include Clostridium and Peptococcus, acetogenic bacteria like Syntrophobacter and Syntrophomonas, acidogenic bacteria like Methanocercina and Methanothrix, methanogenic bacteria like Methanobacterium and Methanobrevibacterium. Now let's see the stages of anaerobic digestion process. The first one is the hydrolysis, which is a breakdown of complex insoluble organic matter into simple sugars, fatty acids and amino acids. And then is the acidogenesis. In this, there is further breakdown of simple sugars, fatty acids and amino acids into alcohols and volatile fatty acids. The next one is the acetogenesis. Here there is conversion of volatile fatty acids and alcohols into acetic acid, carbon dioxide and hydrogen. Finally, methanogenesis. In this, the acetic acid and hydrogen is converted into methane and carbon dioxide by methanogenic bacteria. So you can see the overall process in this chart. The complex organic matters like fat, carbohydrate, protein, lipids goes through a series of steps like hydrolysis, acidogenesis, acetogenesis, and finally methanogenesis, and they are converted into methane, carbon dioxide and water by methanogenic bacteria finally. The use of household digester is not widespread. However, in rural parts of developing countries, anaerobic digesters provide a valuable source of cooking gas. In India and China, household-scale digesters are common. Feedstocks include animal, human, and food waste. Let's see what are the sources of feedstock. Feedstocks are normally biodegradable waste, but energy crops can also be used. Common sources include municipal, commercial and industrial food waste, agriculture waste, wastewater and sludges from industrial waste treatment, food or beverage processing waste, energy crops like maize, grass and silage. The overall process of anaerobic digestion includes pre-treatment, digestion processes, digestate storage, biogas storage, and finally biogas cleanup or upgrading. Pre-treatment is necessary to ensure process efficiency, maximize product yield and reduce operation cost. It is used to remove non-biodegradable materials and homogenize the feedstock. There are several different types of pre-treatment methods, which are physical, mechanical, thermal, ultrasound, electrochemical, and the chemical treatments like alkali, acid, and oxidative, biological, which includes microbiological and enzymatic, and combined process of extrusion and thermochemical. Next, the overall digestion process are characterized by the following. The feed, which includes a wet and dry content. The flow can be batch and continuous. The process stages can be simple or multiple, and the temperature can be mesophilic or thermophilic. And in the case of wet feed, the dry solid should be less than 15% and the waste is macerated prior to processing. And the examples include food waste, manure, slurry. And when the feed is dry, the dry solid content should be higher. It's around 15 to 40%. There is no water addition. There is less mechanical treatment. And the examples include green waste and energy crops. And the digestion process based on batch and continuous. In batch, the digestion process or a stage of the digestion process is allowed to start and finish in a simple vessel. And once complete, the vessel is emptied and the process is restarted with new feedstock. A series of vessels may be used to overcome peak and troughs in flows of feedstock and gas production. Whereas in the case of continuous, the feedstock flows through the plant continuously and it avoids the need to empty digesters and restart the process. It's a labor-intensive and time-consuming process. In continuous, biogas generation tends to be more consistent, although biogas generation rates may be lower than for batch process. And the temperature in mesophilic, it operates at 30 to 40 degrees Celsius. It's a stable process and is suitable for slurries and industrial and commercial food waste. Its most popular option in developed countries, and its retention time is between 15 to 30 days. And in the case of thermophilic, it operates at 50 to 60 degrees Celsius. It's more complex and less stable. It's suitable for a wider range of feedstocks. It's more expensive due to higher energy input requirement. And here the retention time is between 12 to 14 days. And then there is single stage process and multi-stage process. In single stage, digestion occurs in a single reactor. The plant design is simpler and more economical. It produces less biogas and the feedstock takes longer to digest. In multi-stage process, 2 or more reactors optimize the process and it helps to further degrade feedstock and extract more biogas. The process monitoring is another important step, and you should monitor the pH, carbon is to nitrogen ratio, methane, volatile fatty acids, alkalinity, ammonia concentration, trace element, and retention time during the anaerobic digestion process. The process of anaerobic digestion breaks down organic matter to release a methane-rich gas known as biogas. The remaining organic matter is a valuable fertilizer known as digestate. And this image shows the overall anaerobic digestion process. The feedstock after pre-treatment is introduced into the anaerobic digestion digester, and it goes through a series of steps like hydrolysis, acidogenesis, acetogenesis, and methanogenesis. Finally, biogas and digestate are released. The biogas can be utilized for electricity, heat, and it can be used as vehicle fuel, and the digestate can be used as fertilizer and soil improver. Now let's see what's happening in the step methanogenesis in detail. Methanogenesis is the biological production of methane from either carbon dioxide and hydrogen or from methylated organic compounds. Process of methanogenesis was first demonstrated over 200 years ago by Alessandro Volta. The process is strictly anaerobic. All non-methanogenic bacteria belong to archaebacteria. They are also called methanogens. A variety of unique coenzymes are involved in methanogenesis. Methanogens can be classified as chemolithotrophic methanogens and methylotropic methanogens. Methanogenic bacteria are extremely oxygen sensitive. Methanogens require anaerobic conditions. They are found in the digestive system of herbivores, marshes or lake bottoms, all sorts of mud sediments and in man-made anaerobic digesters in sewage treatment plant. Methanogenesis or biomethanation is a form of anaerobic respiration that uses carbon as a terminal electron acceptor, resulting in the production of methane. The carbon is sourced from a small number of low molecular weight organic compounds such as carbon dioxide, acetic acid, formic acid, methanol, methylamines, dimethyl sulfide, and methanethiol. The two best described pathways of methanogenesis use carbon dioxide or acetic acid as a terminal electron acceptor. You can see that the carbon dioxide plus hydrogen will give you methane plus water, and also acetate is broken down to methane by methanogenesis. It is also a type of anaerobic respiration. The production of methane is an important and widespread form of microbial metabolism, and in most environments it is a final step in the decomposition of biomass. During the decay process, electron acceptors become depleted while hydrogen, carbon dioxide and light organics produced by fermentation accumulate. During advanced stages of organic decay, all electron acceptors become depleted except carbon dioxide. Methanosarcina barkeri is a classic and representative species for those methanogenic bacteria that utilize acetate, methanol and methylamine as substrates. Formation of methane from acetate is a oxidoreduction process.